Hi students, this is Mr. Gray. Today we are going to talk about enzymes. So pictured here you can see some enzymes, more of a cartoon picture down here on the right hand side. You can see what is representing the enzyme here in the light blue and what we're going to learn about the substrates and how this all works. Up here you have more of a molecular point of view and I think it's important for students to see these structures so you understand there's not just like these simple structures that we're talking about. There are indeed complex structures and a lot of different chemical bonds between these molecules holding this all together. So enzymes, what are they? So basically they are mostly proteins and they act as biological catalysts. And they're essential for the functioning of the cell. So a catalyst is any substrate that increases the rate of a chemical reaction without being changed or used up. So within your cells, you actually have a lot of catalysts that are going to help increase the rate of the chemical reaction. So some enzymes are RNA, however, most of them are actually proteins. So ribozyme is an enzyme of an RNA enzyme that actually joins amino acids together in the ribosome, so it helps build those peptide bonds. So here you can see the structure of the enzyme down here in the blue, and the portion here where this little object here, we call that the substrate. Substrate's what the enzyme is acting on, is going to nicely fit into that active site. And when it does, it's going to allow that chemical reaction to occur and that molecule or the substrate to change. So we call this model the induced fit model. Enzymes reactions depend on the physical fit between those enzymes and the substrate. So certain enzymes and certain substrates fit together nicely while others don't. So a substrate is a reactant that is going to be catalyzed. And the location where the substrate attaches is, again, it's called the active site. So here you can see the shapes indicated here will fit nicely with the active site on this enzyme. And when they come together, this is actually called the enzyme substrate complex. And in the end, there can be new products that are created from this reaction. So the enzyme acts on the substrate and it creates a new product. And notice the enzyme hasn't changed. It's still there. So it's not used up within the chemical reaction. So the function of enzymes is really going to be determined by the shape of that enzyme and the shape of the molecules that it's acting on. We call that the substrate. So the amino acid chain actually determines the shape since it's made mostly of protein. Those amino acids make up those proteins. So it is going to be the chain of amino acids and how those amino acids are arranged that's going to determine the shape of that enzyme and then in turn its actual function. So here you can see this molecule of peroxide being acted upon by this catalase enzyme. And this may be a lab that you'll be working with in class where we take the catalase and extract it from a potato and we act upon this substrate peroxide and we get the byproducts of that, which are hydrogen and oxygen. Okay, so many times when we look and talk about enzyme activity, you'll see on assessments, you'll get a picture of this diagram. One of the words you see over there is activation energy. So it's important to understand that chemical reactions can still occur without enzymes within your body. However, it's going to take a lot more energy for them to occur and usually a lot more time too. So the amount of energy needed for the chemical reaction to occur is actually called activation energy. So for a reaction to be carried out, let's think about a reaction of like lighting a paper on fire. in order for that to occur, a certain amount of energy has to be present in order to get that reaction started. Right, so we can think of activation energy as being the energy required to start that reaction. So activation energy can actually be lowered with the enzyme. So the enzyme's job, if you can see, without the enzyme, the amount of energy required for that chemical reaction to occur requires a lot more energy based on this graph in order to carry out that chemical reaction. Now, if you look at the reaction with the enzyme, still requires some energy. However, it's much less energy. So having that enzyme present is really important for living organisms in order to carry out those chemical reactions. So in our body, we have a number of substrates, and they are molecules a particular enzyme is going to work on. So there are some examples. Sucrase, and it breaks down sucrose. So whenever you eat table sugar, you have an enzyme that actually breaks down the sucrose, and that enzyme is called sucrase. Another enzyme that's important is amylase, and that breaks down starch. So as you can see, most enzymes end with ASE. In this chart, you can see a number of different types of enzymes that we have for carbohydrate digestion, protein digestion, nucleic acid digestion. Notice that many of them actually end with ACE, but not all of them do. Pepsin and trypsin do not end with ACE. However, they are still really important enzymes. You can see where they're produced. It's a really interesting chart to kind of look at where these molecules are produced and what they carry out, what their pH level is within your body. So reaction rates for enzymes do vary depending on a variety of situations. And these situations are heavily tested typically, so it's really important that you take some time to really internalize this information and understand what we're talking about. So all enzymes have an optimal temperature and pH, and that is where they work best. So when you see that word optimal, some kids get tripped up on that word. Optimal means best, so it's the temperature that's best for that particular enzyme. Or pH for that matter. So whenever the enzyme is working best, it's in its optimal condition. Drastic changes can denature the enzyme. So we talk about denature, it's really breaking down the protein. One way I kind of like students to visualize this, I think about cooking an egg. When you first put the egg in the pan, it's kind of translucent. But over time, as those proteins start to break down, they become more opaque in color. And you can actually kind of visualize those proteins denaturing right in front of you. So the denaturing is the change in the shape of the protein. So enzymes become denatured or change shape with changing temperatures and pH. So if the substrate concentration increases, the rate of reaction increases. It was a limiting factor. So there's other things that actually influence the reaction rate, and that is substrate concentration. So that's what we're talking about with this bullet point here. I kind of just jumped into that really quick. So if the substrate concentration goes up, so that means you have lots of stuff for the enzyme to work on, the rate of the reaction is going to increase because if it was a limiting factor. So if it was a limiting factor, meaning if the enzyme was working faster than it actually had available substrates, then if you added more, the reaction rate would increase. But it's only going to increase up until a certain point. That would be the fastest that enzyme could actually work. So if the enzyme concentration increases, the rate of the reaction can also increase if it was the enzymes that was limiting. So this is actually a pretty simple concept. So if you imagine the enzyme having a job, and its job was to, let's say, organize, let's say, since it's the season when I'm recording this, I'm talking about getting some corn, like shucking the corn, taking corn out and cooking it, right? So if that's your job, you're going to do it as fast as you can. You'd be the enzyme acting on the substrate or the corn. And as that corn was used up, if you ran out of corn, the substrate in that case, you couldn't work any faster, right? You didn't have any corn. So that would be the limiting factor. On the other side of that, if you had more corn and it was just a big, huge dump truck full of corn, and you couldn't possibly keep up with the amount. All right, so long-winded example, but I hope it helped you understand a little bit about how concentrations can actually become limiting factors, and also how you can adjust those concentrations to change the reaction rates. So looking at these graphs, I want you to take a second. I'll pause the video and look at the graphs and determine what the optimal pH is for the enzyme pepsin, what the optimal pH is for trypsin, and if pH is 6.3, which enzyme would work best. So go ahead and pause it. Okay, now that you have had some time to answer those questions, let's take a look. So when I look at this problem, we're looking for optimal, or the best pH for the enzyme pepsin. So when we look at this right here is the relative activity. So if it's working faster, it's going to be higher up here. So it's going to be, the enzyme is going to be producing more products. So looking at pepsin, the peak, or where it is working best, is right in here somewhere. So if you look down here, somewhere between 2 and 4, probably around 3 would be the best answer for pepsin. Now looking at the optimal pH for trypsin, come over here, this is trypsin. Trypsin, you drop down that range, and you're somewhere like a 7.8 possibly at the peak as you drop that down. Right, so it's pretty simple, but this is a very common type of assessment question that you could see. So last one is, if the pH is 6.3, which enzyme would work best? So if we're looking at 6.3, pH 6.3, my mouse disappeared for a second, there it is. pH is 6.3, which one would be working best? 6.3. Well, notice that pepsin is not even functioning at all in a pH of 6. So the answer here would be trypsin. Trypsin, although it's not working at its optimal rate, it is still working much better than pepsin. Pepsin's not even on the charts in this range. So one activity that you will have to be able to do is to label this diagram and identify the model name. So please pause the video, take a second, and label this diagram. Okay, great. Now that you've had some time to practice the labeling, this is something you can expect to be able to do on your assessment. So make sure you understand each component that you have listed here and take some good notes on it. So looking at this, you can see that over here, this is our substrate. This gray thing is the enzyme, and these little locations here are called the active sites. So the substrate entering the active site of the enzyme is the action that's taking place. And here you have the enzyme combined with the substrate, so that's the enzyme substrate complex. And that continues for both of these. Here you can see that the reaction is taking place. And finally, we have the products leaving the enzyme. So this model is called the induced fit model. The induced fit model. All right. So that was your bell ringer. So if you happen to miss the bell ringer, this is your bell ringer for the day. So make sure you get that done if you miss class that day. As always, it's always a good idea to make sure you take some time to go back over your notes, highlighting some specific key terms that we covered throughout this presentation. Remember, vocabulary is key. Reviewing your notes on a regular basis and not just before the assessments is going to help you retain the information much longer. Hope this helps. Have a good day.